Today is a snowy morning, not too cold, I suppose rainy as well, icy and disgusting out there. Today's discussion is over Chapter 7, which is called The Control of Microbial Growth. And there are basically three ways that we can think about control of microbial growth. We can think in terms of the various chemical and physical properties of the things that are involved in the control of microbial growth. These could be the chemical structure of disinfectants or the physics of ionizing radiation. Alternatively, we can consider the strengths and weaknesses of different types of approaches to controlling microbial growth. And so this could be the types of organisms that are affected or the types of conditions that interfere with the action of the various approaches that we're taking, such as the presence of large amounts of organic material might interfere with the ability of the substance to actually impact, for example, the bacteria that you're trying to get rid of. Or alternatively, we can think in terms of general principles. Now, my philosophy in teaching microbiology has been to simply go out of my way to avoid delving too deeply into the chemical and physical aspects of the various mechanisms or approaches that we use to control microbial growth. So the structures of the various chemicals, I mean, they're important, but how much detail do we have to go into to understand these structures? My opinion is that for microbiology, at a general introductory level, the actual structures are not hugely important. Sometimes we'll make a point with the structure of whatever it is that we're working with. But in terms of attempting to memorize these structures, I mean, that is just something that we don't need to do. In terms of strengths and weaknesses, this is something that the book goes into to a great extent. This is something in my actual lecture on this material I'll be delving into. And that is important. I mean, if you're going to be using some kind of means of controlling microbial growth, I mean, it is really important to have some sense of what the strengths and weaknesses of the approach you're using. You know, what will it work against? When will it work? How long does it take for it to work? Et cetera, et cetera. So what I'm going to talk about today is general principles. And so the general principles that we are going to be most concerned with can be described as exponential death. Now, before we get to exponential death, there is a difference between an agent that is bactericidal or microcidal versus something that is a static agent, bacteriostatic, for example. A bactericidal agent actually kills whatever it is that is being treated, whereas a bacteriostatic agent is something that simply inhibits the growth. Now, if you inhibit the growth, you may eventually result in the organism dying. But these ideas of bacteriostatic, bactericidal is something that we'll be delving into in more detail when we get to talking about antibiotics. Antibiotics are also a means by which one can control microbial growth, of course, but the chapter on control of microbial growth in this book is not really focusing on antibiotics. It's focusing on what can be described as less selectively toxic approaches to controlling microbial growth. By selectively toxic, it means that it's toxic to some things and not toxic to others. Ideally, you're working with something that is going to not be toxic to you or your patient, but will be toxic to whatever it is you're treating, such as the bacteria that you're attempting to remove from a surface. But though not all chemical and physical means of inhibiting microbial growth are necessarily toxic to us or patients, antiseptics, by definition, are something that you can use without really hugely harming the individual that you're treating. But it is the nature of antibiotics, particularly, to be selectively toxic. When you're talking about disinfectants or physical means of destroying microorganisms, usually you're working with something that also has a fairly decent capacity to harm you as well. And so those are the kinds of things that we're going to be talking about today, and particularly with emphasis on bactericidal or microcidal agents, virucidal, fungicidal, and in terms of particularly exponential death.